Hello and welcome back to your Chem 4 lecture. We are moving on in chapter 13, covering the last half of the chapter on the chemical properties of monosaccharides and moving into disaccharides and polysaccharides by the end of the chapter. So first, we're going to start by wrapping up our discussion of monosaccharides by talking about some of the chemical properties and the chemical tests that you might do in the future for monosaccharides. So one thing we want to point out here is that while carbohydrates are known as kind of energy providing molecules in your diet, if you're paying attention to your diet, you know that carbohydrates, excess energy from carbohydrates will be converted into fats in your body. I know it's a complicated process. I'm not really a nutritionist or anything like that, but that's the basic kind of level of it. You know that there's also 0-calorie sweeteners, and a lot of these 0-calorie sweeteners are actually based on sugar molecules. You see the flavor of sugar is based on the shape of the molecule interacting with your tongue. So if you change the shape just slightly, you still interact with the sweet receptors, but then if you change the chemical properties enough, you won't be able to process it the way that you do a normal sugar. And that brings up the sugar alcohols here. So for example, sorbitol, xylitol, and mannitol are all examples of sweeteners that you might see on like a sugar-free gum, for example, which is what this is here. Yeah, these sugar-free products have, a sweet taste, yet we can't process them like normal carbohydrates. And we'll talk about how those are prepared now. Okay, so starting with monosaccharides here. So this is D-glucose here, which should look familiar from our previous lecture. Now, interestingly, this is going to involve something from the previous chapter on 2-hydroxyl aldehydes here. You see, all of these aldoses have an aldehyde with a hydroxyl group next to it. So that means that they are positive for the Benedict's test. So if you were to mix the D-glucose here with the copper sulfate solution, copper 2 sulfate solution, you would end up with copper 1 oxide. And rather than glucose, you would have what's called gluconic acid. In other words, the aldehyde would be oxidized to a carboxylic acid. So you can make these sugar acids from the oxidation of an aldehyde form. So one of the aldoses, basically. And the way that you name it then is you get rid of the ose and replace it with onic acid. Yeah, now because this can be oxidized, so any of these aldoses can be oxidized if they have a hydroxyl group next to the aldehyde. We call it a reducing sugar. That comes up sometimes actually in medical situations, so that might show up in the future. So just so you know, any aldehyde, so aldose, with a alcohol group on the second carbon, so most aldoses, is a reducing sugar. Okay, now fructose, on the other hand, has a ketone, and that usually will not be oxidized. However, if you prepare a basic Benedict solution, you can actually rearrange the ketone into an aldehyde. And what happens basically, you make an aldehyde on carbon 1, and what was a ketone becomes a hydroxyl group. So this is basically a way of converting fructose into glucose, just in a basic solution. So while it would fail a normal Benedict's test, if you had a basic Benedict's test solution, you actually would get a positive test for the Benedict's. For the Benedict's test. Again, this rearrangement looks like this. Basically, you take off a proton from the final carbon here, the first carbon, and then this ketone is able to rearrange and become aldehyde on carbon 1. Okay, so that's that. Now, the other way, and this is something that you come up with more often, is these monosaccharides can be reduced as well. Because they have a carbonyl group, the C double bond to an O, that means any of those could be turned into a C single bond to an O. So for example, if you take glucose and you reduce it, you're going to end up with an hydroxyl group, which looks like this. This hydroxyl group here, taking the place of the aldehyde, also means that no longer will the carbohydrate be processed in your body as a carbohydrate. Instead, it'll just taste sweet, but you won't have the issues with calories that you might think of. These sugar alcohols are known as alditols, So this was what we were looking at on the first page, the first slide, sugar alcohols. And this is also known as sorbitol. Yeah, so the way that you name the sugar alcohols is you just take the "-ose" ending and replace it with etol. And you still have to say D or L, since all the sugars that we can process are D sugars. That means that, of course, all the sugar alcohols that we're going to use are D sugars, or D sugar alcohols. And yeah, these are often used as sweeteners in sugar-free products. And there's been, a lot of development in this. This is why the soda companies keep coming out with new, zero-calorie drinks. They're playing around with the new sweetener technology to kind of get whatever tastes the best, Okay, so show me what would happen if you took mannose and you reduced it. So remember, if you reduce it, you're going to turn it into a sugar alcohol. So mannose will become mannitol. So go ahead, pause the video here. Show me what would happen if you reduced it. Draw it in your notes, and then we'll take a look at the answer when we come back. So go ahead, pause. Okay, let's take a look at the answer. So what happens is basically what was the aldehyde up here is going to be reduced to a primary alcohol, CH2OH, and D-mannose becomes D-mannitol. Okay, now how does this relate to the health profession? Well, glucose testing is really important, as we've talked about. We've got hypoglycemia and we've got hyperglycemia that we need to worry about. And so blood glucose can be a really important piece of data when you're analyzing someone's health. So blood glucose usually flows through the kidneys and then it becomes reabsorbed into the bloodstream. If the blood level exceeds 160 milligrams of glucose per deciliter of blood, the kidneys won't be able to reabsorb all of it and it'll spill over into the urine. This is known as glucosuria. So you end up with urine that contains sugar in it. So that can indicate, problems with your sugar processing system. So the way the clinical test works is basically to analyze your urine, you put a paper strip that contains 2-methylalanine and 2 enzymes. It's got glucose oxidase and peroxidase. So take the glucose with oxygen in glucose oxidase, that's going to turn into the gluconic acid, and that's going to produce hydrogen peroxide. Now, if you have this hydrogen peroxide with a dye like 2-methylalanine, then in the presence of peroxidase, it's going to turn into a colored product. So you can immediately see, based on the concentration of the hydrogen peroxide that's produced, which is related to the concentration of the glucose that's produced that's in the urine, you can actually see kind of the levels of glucose in your urine, which is, again, not where it's supposed to be. So this is an example, don't worry about this, you don't have to memorize this or anything, but the intensity of that color will tell you the glucose level. So starting from blue is 0, all the way down to dark brown, which indicates a real problem with glucose in your urine. Okay, so now we're going to move on to linkages of monosaccharides. So far we've just talked about these individual units of monosaccharides, and as we're about to find out, oftentimes they're actually combined into 2 or even more units. So starting from disaccharides, and one of the most common disaccharide compounds, I guess there's sucrose, table sugar that you're familiar with. Lactose is another example of it. And all it is a linkage from one monosaccharide to another. Okay, so again, a disaccharide is just 2 monosaccharides that are linked together. And we call this a dehydration reaction when these 2 monosaccharides link up. It's called a dehydration because it actually produces water. So you take glucose and glucose, you produce maltose, and that leaves you with an extra H2O molecule. A glucose unit and a galactose unit can produce lactose, and then a glucose and a fructose unit produce sucrose. These are the most common disaccharides, and they're kind of the most important ones that we'll be talking about in this class. So maltose is known as malt sugar. So it's kind of older, but I'm trying to think. There's some. I know there's some like dessert places that still use malted sugar. It has a very particular taste to it that sometimes you like. I know, like for my grandpa, he would like go to the ice cream parlor with us, and he would order a malt, which actually I don't know, I think that tastes pretty good too. But it is like it seems like it was popular in the 1950s or something like that. So so a malt milkshake is a milkshake that uses maltose as part of the ingredients. And again, it just offers like a different flavor. What else is there? There's also Whoppers. You ever have Whoppers, the little malted milk balls? They have maltose in them as well. So just a slightly different flavor. And maltose itself is composed of two alpha D glucose molecules. And this can be made by the hydrolysis of starch. Yeah, okay, so it's right here. It's present in cereals, candies, and then it's also famously used for brewing. So like brewing different beers, maltose is one of the best sugars for that because the yeast that you use to brew survive really well in maltose. And there's two different forms of it. There's an alpha form and a beta form that we're going to talk about on the next slide, I hope. Yes, okay. Okay, now here's the main thing that we want to understand about these disaccharides is that the linkages on di and polysaccharides are what's really important. So what happens here is for maltose in particular, you take an alpha D glucose and another alpha D glucose. Now, what's going to happen is there's a dehydration reaction where this OH group and the hydrogen of this are going to leave as water. And that's going to leave us with just an oxygen that's going to connect the two molecules. That's what we can see down here. That's why we call it a dehydration. It's because we're losing water, is the way that you can think about it. Now, what's important to understand is we can classify this linkage because the first molecule is linked from carbon 1 here, if you look on its Haworth projection, to the second molecule on the fourth carbon here. We call this a alpha 1,4 glycosidic bond. Now it's called alpha because, of course, this OH is below the plane of the Haworth structure. Since it's below the plane of the Haworth structure, that means that it is alpha, right? If it were above, then it would be beta. Now you form a 1,4 linkage here, and that produces your maltose. And then this maltose can either be alpha or beta as well, because it still has this opening over here, where it'll start as an alpha based on the linkage of these two units, but this side can still flip into beta. So that's what we want to understand from there. Now, lactose, on the other hand, is starting from beta D-galactose and alpha D-glucose. What happens here is we have a beta 1,4 glycosidic bond. In other words, it has to be the beta from carbon 1 bonded to the fourth carbon on the other one. That's where we have this oxygen kind of bridging in between. I think we have a better look at it here. So again, we have the hydroxyl group on carbon 1 of a beta D-galactose forms a glycosidic bond. That's this O here. This is what we call this glycosidic bond, is just this oxygen that bridges the two monosaccharides. And that links to carbon 4 of the alpha D glucose. Oh, and notice here, again, we have carbon 1 on the other side of this glucose molecule. That could be alpha or it could be beta. Yeah, so again, we call this a beta 1,4 glycosidic bond because the OH group of carbon 1 of beta D-galactose is the one that forms the glycosidic bond. Okay, the last disaccharide that we're going to really worry about here is sucrose. And sucrose is an alpha-beta linkage, actually. You have alpha D-glucose and beta D-fructose. So it's actually an alpha beta 1,2 glycosidic bond. That recognizes that we have the alpha form of glucose and the beta form of fructose, and they're going to form a glycosidic bond. Again, that's just where you have oxygen connecting the two units. So now the reason that we have to say both alpha and beta is because we have to have the carbon 1 of glucose in the alpha form where the OH is on the bottom, and we have to have the beta form of fructose where the OH is on top. Otherwise, we get a different type of linkage. Yeah, okay, so this is a better look at it here. again, from carbon 1 on the glucose to carbon 2 on the fructose. Now, because carbon 2 on the fructose, that's the one that's the ketone nominally in the Fisher projection. That's the one that actually is, alpha or beta in the closed form, the Hayworth projection. Okay, so let's take a look, and we'll just briefly talk about sweetness and what that means. So sugars and artificial sweeteners have different flavors, and they differ in how sweet they are. So the way that we assign the flavor is we say that sucrose is a value of 100 sweet units. And compared to that, we can see that monosaccharides like lactose and glucose, I'm sorry, I think this should be glucose here, are less sweet than sucrose. Fructose, on the other hand, is 75% more sweet. Okay, now disaccharides, lactose and maltose are less sweet than sucrose. And then when we get to sugar alcohols, they're 60%, 80%, and then about 100% is sweet. So xylitol is right at about the same sweetness level as sucrose, even if it tastes a little different. Now, non-carbohydrate sweeteners are what we're going to talk about, right? So we had sugar alcohols here, disaccharides, monosaccharides. The structures aren't necessarily related at all. So stevia, aspartame, saccharin, sucralose, neotame, and advantame. I think the most popular one right now is stevia. I feel like I hear that one a lot. Aspartame has been popular in the past. I think that was the first no-calorie sweetener that was used. Saccharin and sucralose, I know I've seen as well in recent times. And we'll take a look at what those structures look like here. So sucralose is what's known as Splenda, and it looks like it's a sucrose molecule, but with the exception that some of the hydroxyl groups have been replaced with chlorines. So you see there should be an alcohol here. Well, that's been replaced with a chlorine. Should be an alcohol over here. That's a chlorine, and another alcohol replaced by a chlorine up here. So again, the shape is very similar. So our tongues are full, yet our body can't process it. So it's not going to be, the energy of the bonds isn't going to be harvested. Okay, next up, aspartame. Aspartame, as you can see, is totally different. It's an amine and a carboxylic acid, and an amide in here in the middle, and there's a benzene ring over here. This is what's known as NutraSweet. And it's, again, totally non-carbohydrate. It's actually made of amino acids. So there's aspartate and the ester of phenylalanine. So again, it's non-carbohydrate. You don't have to memorize the structure or anything. Just kind of want to point out, again, this tastes sweet to us for whatever reason. Okay, so all of that, let's go ahead, check your notes here and tell me which monosaccharides are involved in the following. Lactose, choose the two that are there, maltose, and sucrose. Tell me which monosaccharides are involved in each of them. Pause the video here, take a look at your notes, and when you come back, let's take a look at the answer. So pause. Okay, so let's take a look. Lactose is a combination of alpha-D glucose and beta-D galactose. Maltose is a combination of 2 units of alpha-D glucose. And sucrose is a combination of alpha-D glucose and beta-D fructose. And again, normally memorize these, but of course, if you have them in your notes, it's an open note test. So just put them in your notes. Okay, all right. So now how does this link to kind of everyday health settings? And there's actually a really important consequence of saccharides for blood, which is that our blood types are based on the saccharides that are like tagged units on our blood cells. So type O is what's known as the universal donor, because type O has basically the standard terminal saccharide, where it's a link of, here, I got to look them down here. It's N-acetylglucosamine, galactose, and fucose. So those three in that line, as you can see, that's the same that is in type A and the same that's in type B. The only difference is type A and B have these other two, N-acetylgalactosamine and galactose units that are kind of sticking out there. Now, because, so I think let's go ahead. So we've got O with 3 common terminal ones. A is the same, but with N-acetylgalactosamine. And B has the same, but with a second molecule of galactose. And then AB type would consist of both of the monosaccharides from types A and B. Okay, so you don't need to memorize these structures, and you don't even need to put them in your notes. Just want to point out here the structures are different for each of them. Okay, now the way it is here is that type O blood, if they're given type A and type B or type AB blood, they will produce antibodies against it. Basically, your body is used to this tag. So if it sees something that's different from this tag, then it knows that something's wrong. So if you added in type A, it says, oh, hey, there's this N-acetylgalactosamine, that's in the wrong place, I'm going to attack that blood. Same thing here with the galactose. It says, hey, there's an extra galactose, that's not my blood, I'm going to attack it. However, take a look, type O blood, because it has these three here, these three are present here in A and here in B. So there's nothing extra in the type O blood that the type A's and the type B's wouldn't recognize. That's why type O is universal donor blood. Now, on the other hand, if you are a type AB blood person, that means that you are fine with type A blood because you have the type A terminal saccharides, and you're fine with type B blood because you have the B terminal saccharides, and of course you're fine with type O blood because it can be a universal donor. So yeah, that's no problem, right? You can, if you're type AB, you can accept all kinds of blood. Okay. Go. Okay, there we go. All right, here we go. So melibiose is a disaccharide that's 30 times sweeter than sucrose. Ask yourself, what are the monosaccharides in melibiose? This is a good time to look through your notes and try to figure this one out. Then ask yourself, what type of glycosidic bond links are involved in or what type of glycosidic bond links these monosaccharides? And then look at the structure and ask yourself, is this the alpha form or the beta form of melibiose? This is, again, an open-ended question, so please pause the video here, take a look through your notes, and then we'll explore what the answers are here in a little bit. Again, we didn't talk about these specifically before, but if you can identify these monosaccharides by looking back at your monosaccharides notes, then you should be able to identify the other types here, the glycosidic bond that forms and alpha or beta form for this one. So pause the video here, and we'll take a look at the answer when we come back. Okay, let's take a look. So what are the monosaccharide units? Well, the first monosaccharide unit, check out the OH group on carbon 4. So we can see that there's only 3 possible ones I'm going to ask you. Okay, it's going to be glucose, galactose, or fructose. Those are the only 3 that are possible for us. Here for carbon 4, the OH group is sticking up. And when OH is pointed up, that's a pretty good indication that it's D-galactose. If it were pointed down, then it would be D-glucose. Next up, we look at carbon 1, and we can see that it's the OH would be pointed down. So that's the alpha form of galactose. So alpha D-galactose. Okay, next up. Here, the OH group is pointing down for melibiose for the second disaccharide, or the second monosaccharide. When it's pointing down, that's glucose. So we have a unit of alpha D-galactose, we have alpha D-glucose over here. Now, what about the bond type? Well, it's an alpha carbon from the galactose here, right? So we know it's alpha, and then it's a link between the 1 carbon over here and the 6 carbon over here. So it should be an alpha 1,6 glycosidic bond. Okay, finally, you look down here at melibiose, we see that the other carbon 1 over here is pointing down with its OH group. So that's the alpha form of melibiose. Okay, so that's how we determine it. Okay, so we have just a very brief section in your chapter on polysaccharides, but we want to make sure we talk about them to understand because they are important, even if even if there's not that many questions that you're going to see about them necessarily. But what polysaccharides are basically just when you take a monosaccharide and you join it together with another, and then you take that one and join it together with the next, with the next, with the next, with the next, until you get many, many, many monosaccharides in a long chain. Some of the ones that you may have heard of before would be cellulose, amylose, and amylopectin are all examples. And these are all actually polymers of alpha D-glucose. So alpha D-glucose, again, super important for us to be able to process energy. And the reason for that is that plants developed a way to make alpha D glucose really efficiently through photosynthesis, and then they just started building with it after they got that. So amylose is a straight chain polysaccharide, and all of the glycosidic bonds are the same. They're all alpha 1,4 glycosidic bonds. So amylose is unbranched here. Now, amylopectin is related. It has a lot of alpha 1,4 glycosidic bonds, but it can also branch out. It also forms alpha 1,6 glycosidic bonds to form a branch like this. So amylose is about 20% of the polysaccharides that are involved in starch, and it consists of 250 to 4,000 of these monomers, these alpha D glucose monosaccharides. And again, they're all connected by alpha 1,4 glycosidic bonds. We call it a straight chain polymer. Now, it is actually coiled in a helical fashion, but because we would draw it in a straight chain in its Haworth projection, it's considered straight chain in that way. We don't mean the three-dimensional structure necessarily, just the linkages are all the same. Amylopectin, on the other hand, makes up the other 80% of starch, and this is a branched chain polysaccharide. So these glucose molecules are connected by alpha 1,4, just like amylose, but then also we add on top of it some branches with alpha 1,6 glycosidic bonds. Okay, so these starches will break apart in water. So like you can boil them in water, and that makes them give like smaller saccharides, which are called dextrins. And then finally, they will hydrolyze to maltose and glucose. So even though starches maybe don't taste sweet, our bodies are able to easily convert them into glucose, and then from there, harvest the energy from it. Okay, glycogen, on the other hand, is a polymer of glucose that's stored in liver and muscle areas for animals. It's basically a really convenient way to store energy because it's easy for our cells to hydrolyze it. And that's a way that we can maintain the blood level of our glucose. And in between meals, we can, eat that glycogen up and use that energy when we're not eating. Structurally, it's very similar to amylopectin. So again, that means that it has alpha 1,4 glycosidic bonds, and then also branches of alpha 1,6 glycosidic bonds. But those branches occur more often than in amylopectin. These branches occur every 10 to 15 glucose units. And I didn't really tell you here, but this occurs less often. Okay, so the final polysaccharide we want to talk about is cellulose. And you probably heard of cellulose because it is the main component of the cell wall of plant cells. And as such, it's also the major structural unit of wood. And it's a polysaccharide, no surprise here, right? And it's all glucose units, and it's all unbranched chains with beta 1,4 glycosidic bonds. Now, because it's so highly, because it's fully chained, it will not form hydrogen bonds with water. And so it becomes insoluble in water, just like wood is, of course, insoluble in water. The way that it's structured allows for a rigid structure in cell walls, and that allows plants to have their shape, both when they're alive and then, of course, after they're dead. And because we can't digest these beta 1,4 glycosidic bonds, so that's a big difference here, is rather than alpha 1,4 glycosidic bonds, these are beta 1,4 glycosidic bonds. We don't have the enzymes to break those down. So we can't actually digest. This is just what's known as fiber. So this cellulose is also, for example, in cotton, it's a beta 1,4 glycosidic bond. So you take a beta D glucose unit, and that connects to the next beta D glucose unit, and on and on and on. Okay, so given these two structures, just tell me which polysaccharides are there and which type of glycosidic bonds are involved. So go ahead, pause, just take a look at the previous slides, and we'll take a look at it here. So pause. Okay, so let's take a look. Again, cellulose is the cotton one, and that only has beta 1,4 glycosidic bonds. The potato contains both amylose, which has alpha 1,4 glycosidic bonds, and amylopectin, which has alpha 1,4 and alpha 1,6 glycosidic bonds. Okay, well, that covers our carbohydrate chapter. I know that was a lot of information, but hopefully we're taking good notes. If not, make sure, take some more notes, and have those ready for the test when we're ready for that. Until then, as always, ask questions in the comments down below, and I'll get back to you just as soon as I can. Until then, bye-bye.